we're gonna start in 5 minutes so that people can join in as usual. And we're not gonna discuss anything new, as I mentioned that I was taking revision so that we can understand from where we have started and where we are ending our membrane discussion. Then only we're gonna switch to our next modules. A little update with respect to the lectures. I'm still having certain difficulty in uploading them on the portal, so I've started uploading the lectures on my YouTube channel. I have, I guess, uploaded 10-11 lectures till now. I'm gonna share the link with you guys maybe by the end of this class, or I'll just put it on the in the chat itself, so you guys can go and watch them. If you have any queries, feel free to mail me, or yeah, mail is the best option. I do reply to all the mails. Okay, so we gonna start our revision in 5 minutes. I guess we can start our revision for today. So, in the last class, as you guys remember, that we summarized our lipid discussion. So, I hope that it's pretty more clear. But at the end of the day, I'm gonna suggest you guys to always go back to the books and the videos. Videos are just a way of give you an abstract information about a new topic, and books will give you the comprehensive knowledge which you actually need for understanding a particular topic, right? So we're gonna discuss about proteins. Membranes, biological membranes have lipid and protein, right? So we discussed pretty much about the lipid, their structure, their kind of role. But if you remember, membranes are also functional in nature, right? They have certain functions associated with them. They are not there just for the sake of boundary or for separating the cells internal compartments from the outside, right? They do play a functional role, and those roles are majorly performed by proteins. So proteins are polymers, right? They are big molecules. So to understand how a polymer will behave, act, or will be found in the natural like inside our body, it is important to go back to the units through which they have been made, right? I hope you understand this. For example, if you want to study or understand anything which is happening at a macroscopic level, there is a giant structure which is present, which you can see, right? But to understand how that structure is being formed, at the end of the day, a huge structure is generally made from small subunits of units of things, right? If we know detailed information about these smaller units which combine and make that macromolecular structure, it is easy for us to understand, predict, and maybe analyze, and also imagine what else can be the possibility if we merge monomers with certain properties, right? That is why when we are going to learn about membrane proteins, we should first know what proteins are at the end of the day, right? So proteins are polymers of amino acids, with each amino acid residue joined to its neighbor by a specific type of covalent bond. Proteins can be broken down or hydrolyzed to their constituent amino acids by a variety of methods, and the earliest studies of proteins naturally focused on the free amino acid derived from them. There are 20 different amino acids which are commonly found in proteins, and this statement should not be taken like a line carved on a stone. Why? There are more than 20 different amino acids which are found, but most of them, the proteins which we're gonna study, they are made up of 20 different amino acids, right? There are more than 20 amino acids in the living system, but the proteins, the functional form of proteins, they generally have these 20 amino acids only. Okay, so amino acids share common structural features. So again, as we have talked about the classification of lipids, which is very difficult, right? Because there are different types of lipids which are present in the living system, yet we try to classify them. So at the end of the day, when we are trying to classify something, it indicates that we are trying to bring together like molecules so that it is easier for us to understand and learn, right? So even for the amino acids, though there are 20 different types of amino acids, but all of them can be grouped together in some number of groups because they share some common properties. Those properties, they can be structural property, they can be chemical property, they can be any other property which you can imagine, right? So amino acids share common structural features. All 20 of the common amino acids are alpha amino acids. They have a carboxylic group and an amino group bonded to the same carbon atom, which is called the alpha carbon. Okay, they differ from each other in their side chains or the R group, which vary in structure, size, and electric charge. and which influence the solubility of the amino acid in water, right? So this is the representative way of how a amino acid will look like. Till here we haven't talked about what amino acid. We are only talking about 20 different amino acids which are present in the proteins in our system, and all of these amino acids have this common structure, right? And what is this common structure? They have carboxyl group, they have amino group. To satisfy the valency of the central carbon, they have one H associated with it, and they have another group which always varies. So this is the difference or the dissimilarity which brings 20 different amino acids. Why? Because all these three bonds, the groups which are attached to these three bonds, they are same. So from where we are getting 20 different amino acids? We are getting whatever is associated at the place of R. Okay. So these are the 20 different amino acids which are present in proteins. As you remember, in our earlier discussion also, I mentioned that amino acids can be classified by R group. So here we are classifying on the basis of side chain or the R group. So we can classify amino acids into 5 different groups. They are non-polar aliphatic R groups, aromatic R groups, polar uncharged R groups, positively charged R groups, and negatively charged R groups. Okay. So what are non-polar aliphatic R groups? They are glycine, alanine, proline, valine, leucine, isoleucine, and methionine, right? So there are 7 different amino acids. So there are 7 different amino acids which belong to a group which have non-polar and aliphatic side chain, that is R group. They are glycine, alanine, proline, valine, methionine, isoleucine, and leucine. Okay. Then aromatic R group. Aromatic R groups have side chain which have aromatic structure associated with them. They are phenylalanine, tyrosine, and tryptophan. Then talking about polar uncharged R groups, there are 5 different amino acids. They are serine, threonine, cysteine, asparagine, and glutamine. Okay, they are polar but yet they are uncharged. Then positively charged R groups, we have lysine, arginine, and histidine. Negatively charged R groups, we have aspartate and glutamate. Okay. Now we're gonna discuss about these these classes of amino acid which we have just classified on the basis of R group. Okay. So the first group was non-polar aliphatic R groups. So non-polar aliphatic R groups, the R groups in this class of amino acid are non-polar and hydrophobic, right? The side chain of alanine, valine, leucine, and isoleucine tend to cluster together within proteins, stabilizing protein structure through the hydrophobic effect. Now, during the beginning of this discussion, as I mentioned, if we know the properties of subunits through which the polymer is made, we can actually analyze and predict the behavior at the macroscopic level. Correct. So when I was mentioning that on the basis of side group or the R group, there are 5 different classes of amino acids. There are 20 amino acids at the end of the day, but these 20 can be divided further into 5 classes so that we can understand better what properties are associated with them. So the first group which we are discussing is the non-polar aliphatic R group. As the name suggests, these amino acids are non-polar and they have carbon chain associated with them, right? Aliphatics, linear chains. So there are 7 amino acids which are glycine, alanine, proline, valine, leucine, isoleucine, and methionine. So the property which is associated with non-polar and aliphatic R group is that they are hydrophobic in nature. And the moment we hear the word hydrophobic or non-polar, we know that they will prefer to stay in an environment which support their property, and their property is hydrophobic and non-polar. So in the membrane context, most of the time the amino acids which are present inside the membrane or the proteins which are in the membrane and the region of the protein which is inside the membrane will actually have predominantly amino acids which belong to the first class of amino acid which we just classified on the basis of side chain. right? See how things are connected. The moment we understand the property of subunits through which the polymer is made, we can actually get an idea about what function their location might be. It is not 100% rule, because exceptions are always there, and depending upon the functions, some of the properties can be compromised, but as a whole we can actually predict. Correct? So glycine has the simplest structure, although it is most easily grouped in the non-polar amino acids. It is very small, and chain makes no real contribution to interaction driven by the hydrophobic effect. Methionine is one of the two sulfur-containing amino acids, has a slightly non-polar thioether group in its side chain. Proline has an aliphatic side chain with a distinctive cyclic structure. The secondary amino or imino group of the proline residue is held in a rigid conformation that reduces the structural flexibility of polypeptide regions containing proline. Okay. The second group which we discussed, which we just classified based upon the side chain, that is the R group, was the aromatic amino acids. So there are three different amino acids which are aromatic in nature. They are phenylalanine, tyrosine and tryptophan. Okay. With their aromatic side chains, they are relatively non-polar, that is still hydrophobic, right? Now again we can associate this property with the property of a macromolecule, that a protein. Proteins having non-polar aliphatic amino acids as well as aromatic amino acids, they are hydrophobic in nature, indicating that they'll prefer to stay in the membrane or the hydrophobic environment. All can contribute to the hydrophobic effect. The hydroxyl group of tyrosine can form hydrogen bonds and is important functional group in some enzymes. See, now here comes the beauty. If we look at the tyrosine, it is an aromatic amino acid, right? But still it has a OH associated with it. So wherever there is OH, you know hydrogen bond potency is there, correct? So now with these two properties, can we guess where tyrosine is most likely to reside in the membrane? Because we are talking with respect to the membrane proteins, we are not talking with respect to the cytosolic proteins, right? So with your limited knowledge and information which we just shared, can you guess where tyrosine molecule would like to reside in a protein and the protein is present in the membrane? So the answer is pretty simple. As it is hydrophobic, so it will definitely stay in the membrane protein, wherever there is the part of a protein which is inside the membrane. Also it has OH, right, which has a potency of making hydrogen bonds, indicating that it will likely to be in the polar environment, right, because it can make hydrogen bonds. So what will happen is, it has been observed that tyrosine actually prefers the boundary of the membrane, right? So most of the molecule, most of the chain and the aromatic ring will stay inside the membrane, right? But this OH will be protruded outside the membrane, correct? I don't have a representative figure, but let me try if I can make some things. So imagine this is your membrane bilayer, okay? This is your protein. Suppose this is multipass protein, correct? So this part, sorry for the, this part is present inside the membrane, which means it must have amino acids which are hydrophobic in nature, right? And so what are those amino acids which are hydrophobic in nature? It belongs to the class of non-polar aliphatic R group and the aromatic R group, correct? But in the aromatic R group, there is a amino acid called tyrosine which has OH associated with it, right? So what will happen, despite having a hydrophobic nature, the tyrosine molecule will reside here in the protein, because most of the amino acid will be inside the membrane because it is hydrophobic, but the OH can protrude out and hence help in the functionality of the protein, right? I hope you can imagine, or at least like just with this rough drawing try to understand what I'm trying to say. Okay, the tyrosine and tryptophan are significantly more polar than phenylalanine because of the tyrosine hydroxyl group and the nitrogen of the tryptophan indole ring. Tryptophan and tyrosine, and to a much lesser extent phenylalanine, absorbs ultraviolet light. This accounts for the characteristic strong absorbance of light by most proteins at a wavelength of 280 nanometer, a property exploited by researchers in the characterization of the protein. So here you can see that if there are aromatic amino acids present in the protein, which is most likely because it belongs to those 20 amino acids which are present in biological system and the protein, aromatic amino acids will definitely be there. And the property of aromatic amino acid is that they absorb UV light at 280 nanometers, correct? Hence we can actually characterize and classify and identify proteins based on this. In comparison to the three aromatic amino acids, that is phenylalanine, tryptophan and tyrosine, tyrosine and tryptophan are comparatively more hydrophobic, and you can easily see here in the absorption spectra itself. Okay, then the third group was polar uncharged R group. Now from here what we can understand, till now whatever we have discussed, they are the hydrophobic amino acids, right? The non-polar aliphatic, there were 7 amino acids, and then aromatic amino acids, there were 3 amino acids. Now we are talking about polar uncharged R group. There are 5 amino acids to this class. The R group of these amino acids are more soluble in water and more hydrophilic than those of the non-polar amino acids because they contain functional groups that form hydrogen bonds with water. See how the property of amino acid changed just now. Till now we have discussed about the hydrophobic amino acids. Now we are talking about the hydrophilic amino acids. So if we look at our the structure which we just made here, so chances are these polar uncharged R groups, they will prefer to stay at a place where they can make hydrogen bonds. So hydrogen bonds will most likely be in the cytosolic or the extracellular side because that is the aqueous environment, right? So they will be there. This class of amino acids includes serine, threonine, cysteine, asparagine and glutamine. The polarity of serine and threonine is contributed by their hydroxyl groups, and the polarity of asparagine and glutamine is contributed by their amide groups. Cysteine is an outlier because its polarity contributed by its sulfur hydride group is quite modest. Cysteine is a weak acid and can make weak hydrogen bonds with the oxygen or nitrogen. Positively charged R groups and negatively charged R groups. So positively charged or basic R groups or amino acids, they are the most hydrophilic R groups, and those that are either positively charged or negatively charged, the amino acids in which the R group have significant positive charge at pH 7, that is our physiological pH, are lysine, which has a second primary amino group in the position on its aliphatic chain, arginine, which has a positively charged guanidinium group, and histidine, which has an aromatic imidazole group as the only common amino acid having an ionizable side chain with a near neutrality. Negatively charged amino acids or acidic amino acids, the 2 amino acid having R group with a net negative charge at pH 7 is aspartate and glutamate. Okay, so these are just some basic information. As you know, we have already discussed these particular things, but still as a revision, just let me start sharing with you guys a revision video which I have shared earlier also in the slides. 1 minute. Yeah, here it is. We're not gonna watch the whole video, but it's just a throwback to the time when we were actually studying about the amino acids. Okay.
Essential units, called amino acids, bound to each other by peptide bonds to form an amino acid chain. They sort of resemble a string of beads, and they get twisted and folded into a final protein shape. To make a protein, we need to get to know two things: the ingredients, which are the amino acids, and the recipe, or how the finished amino acid chain folds into the protein. Humans use 20 amino acids in our day-to-day protein making. So we have alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, and valine. Phew, that's 20. One way to divide these is into the ones that we make ourselves and the ones that we cannot make ourselves. There are 5 amino acids that are dispensable: alanine, asparagine, aspartic acid, glutamic acid, and serine, because we can make them de novo ourselves at any time, and in good quantity. Then there's 6 of them that we call conditionally essential, because we can make them most of the time, but not always. And these are arginine, cysteine, glutamine, glycine, proline, and tyrosine. Finally, there are 9 of them that we cannot make ourselves: histine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. And as a result, we have to obtain them from our diet. We call these the essential amino acids. Alright, so the amino acid. Just from the name, you can tell they've got an amine group, or NH2, and also an acid, in this case a carboxylic acid group, or COOH. The amine and carboxylic acid groups are both bound to the same carbon, called the alpha carbon. Now, at a physiologic pH of 7.4, the amine group has a positive electrical charge, and the carboxyl group has a negative charge. Having both a positive and a negative charge makes amino acids a type of Zwitterion, which is German for hybrid or double ion. Now, the alpha carbon also has a side chain, sometimes marked as R, and this side chain gives the amino acids certain properties, which can play an important role in the overall protein structure. First, the side chain can be hydrophilic or hydrophobic, so water-loving or water-hating. Hydrophobic amino acids have non-polar side chains. This might be in the form of an alkyl side group, which is a saturated hydrocarbon, seen in valine, glycine, alanine, leucine, isoleucine, methionine, and proline. Alternatively, it can be in the form of an aromatic side group, which involves a 6-carbon ring, like in phenylalanine, tyrosine, and tryptophan. Now, hydrophilic amino acids have polar side chains. These polar side chains might be acidic, like when their side chains contain additional carboxyl groups, like aspartic acid and glutamic acid. Other hydrophilic amino acids have polar side chains that are basic, like lysine, histidine, and arginine. At physiological pH, the acidic groups lose a hydrogen, and the basic groups gain a hydrogen. Finally, some polar side chains are neutral. For example, they can contain hydroxyl groups like serine, threonine, or tyrosine, or sulfhydryl groups like cysteine, or carboxamide groups like asparagine or glutamine. Now, keep in mind that the charge on an amino acid really depends on its side chain, as well as the pH. For example, at a very low pH, the amine group is positive, while the carboxyl group is neutral. And at a very high pH, the amine group is neutral, and the carboxyl group has a negative charge. And at a pH that's somewhere in between, both groups are electrically charged, and they cancel each other out, resulting in no net charge for the amino acid. The just right pH, also known as the PI or isoelectric point, is different for every amino acid, and it depends on the specific side chains. For amino acids to link up in a chain, the carboxyl group of one amino acid has to bind to the amine group on another amino acid, creating a single peptide bond. This is a condensation reaction, meaning that two amino acids are basically smushed together, and the OH from the carboxyl group along with one of the hydrogens from the amine group get released as a water molecule in the formation of an amide bond. While technically being a single bond, it actually has the properties of a structurally stronger double bond, thanks to the property of resonance. Now, resonance is a property of a molecule where electrons get shared across the molecule, while keeping the arrangement of atoms the same. Basically, the electrons from neighboring functional groups of the amino acid are borrowed, and that makes peptide bonds stronger and more stable. So amino acids are, essentially, a carboxyl group, an amine group, a side chain, and a hydrogen, all bound to an alpha carbon. Now, there's an interesting geometric property here called chirality, which means that each amino acid can exist in two forms that look like mirror images of each other. These two forms are called enantiomers of each other. We have the left or levo-oriented amino acids, as well as the right or dextro-oriented amino acids. While similar, these are definitely not the same. Think of it like a pair of shoes. Even though they're made out of the same materials and generally look alike, the left and the right shoe are not interchangeable, at least not without a lot of pain involved. As it turns out, proteins are only made out of levo-oriented amino acids.
From this, I want you guys to ponder upon that why only L amino acids are present, right? In the biological system, if there is a possibility of having both L and R amino acids, L or D form of amino acid basically, then why we predominantly have L amino acids, okay? And if that is the case, then do we have any D amino acid? Is there a possibility that proteins or enzymes which are present in our system, they actually have D amino acid of the 20 amino acids which we are talking about, okay? Think about it. So the overall summary of the amino acid is that the 20 amino acids commonly found as residue in proteins contain an carboxylic group, an amino group, and a distinctive R group substituted on the carbon atom. The carbon atom of all amino acid except glycine is asymmetric, and thus amino acid can exist in at least two stereoisomeric forms. Only the L stereomers of amino acid with a configuration related to the absolute configuration of the reference molecule L-glyceraldehyde are found in proteins. Here is the hint why we have L amino acids, okay? Amino acids can be classified into 5 types on the basis of the polarity and charge of their R groups. Other less common amino acids also occur either as constituent of proteins, usually through modification of common amino acid residues, or as free metabolites. Amino acids vary in their acid-base properties and have characteristic titration curve. Monoamine monocarboxylic amino acids which have non-ionizable R group. Now you need to remember your classification of amino acids to understand this property, right? Hence we actually need classification to understand the basic functionality of macromolecules. Are dry protic acid at low pH and exist in several different ionic form at the pH if the pH is increased. Amino acid with ionizable R groups have additional ionic species depending on the pH of the medium and the R group. Other way of classifying amino acids, as I mentioned pretty earlier, that those 5 different groups, they are based upon the side chain or the R group. Now we can classify amino acids if we just want to classify them as polar or non-polar. So even from here we can get an idea that which amino acids will be present inside the membrane and which will prefer to stay in the polar environment, right? So talking about non-polar amino acids, we have alanine, valine, leucine, isoleucine, proline, phenylalanine, tryptophan and methionine. Polar amino acids include glycine, serine, threonine, cysteine, glutamine, tyrosine, asparagine, lysine, arginine, histidine, glutamic acid, and aspartic acid. This is the similar classification, except other ways we can classify is aliphatic, we have already discussed, aromatic we have discussed, polar, uncharged we have discussed. Take sulfur containing as a separate group, then cations, anions, and imino as a separate group for classification. Now we have talked about the protein amino acids, right? So these are the monomers through which the protein is made. So how a polymer is actually made? Polymer will be made by joining these monomers, right? So how are these monomers joined? So two amino acid molecules can be covalently joined through a substituted amide linkage termed a peptide bond to yield a dipeptide. Such a linkage is formed by removal of the elements of water, a hydroxyl moiety from the carboxyl group of one amino acid, and a hydrogen atom, the amino group of another. Okay, so you can see it's a dehydration reaction. You can see this is the first amino acid having its amino side here. This is the second amino acid with R2. It can be any amino acid, and its carboxyl side is this side. So whenever they come together, there is a dehydration reaction and a covalent bond will be formed. This covalent bond is called peptide bond, right? Another thing which we can take from this is that amino acid or the protein sequence will be read from N terminal to C terminal. N terminal will be the first amino acid which is being joined, and the C terminal will be the last amino acid which is joined in the peptide chain. When a few amino acids are joined in this fashion, the structure is called an oligopeptide. When many amino acids are joined, the product is called polypeptide. Proteins may have thousands of amino acid residues. Although the term protein and polypeptide are sometimes used interchangeably, molecules referred to as polypeptide generally have molecular weight below 10,000 Dalton, and those called proteins have higher molecular weight. No generalization can be made about the molecular weight of biologically active peptides and proteins in relation to their function. To prove that point, we have referred to this table which clearly indicates that the function and the number of peptides has nothing to do with the function of the protein. The molecular weight and the function of the protein are not directly or indirectly related. Okay. Amino acids can be joined covalently through peptide bonds to form peptides and proteins. Cells generally contain thousands of different proteins with each a different biological activity. Ionization behavior of peptides reflect their ionizable side chain as well as the terminal amino acid and carboxyl groups. Proteins can be very long peptide chains of 100 to several amino acids. 1000 amino acid residues. However, some naturally occurring peptides have only a few amino acid residues. Some proteins are composed of several non-covalently associated polypeptide chains called subunits. Simple protein yield only amino acid on hydrolysis. Conjugated protein contain in addition some other components such as metal or organic prosthetic groups. So these were just some basic details about this. From here we switched gear, not to the from the protein perspective, but we actually switched our gears towards how we can identify given the sequence, because now we know we can read protein sequence from N terminal to C terminal. We know the properties of single amino acid, right? So if you have a sequence in front of you, can you actually predict their behavior? So for that we actually scientists at that time, they actually came up with a scale which was called hydropathy scale, right, or hydropathy index, which basically tells you the potency of an amino acid residue to stay either in the aqueous environment or stay in the non-polar environment. So this is the energy associated with that. Then we try to understand, given the hydropathy index, how we can actually say that which part of this sequence which we have right in front of us will most likely be in the membrane. Okay, and then this was the example through which we understood this. After this we directly went to the membrane protein parts. So what we have discussed amino acids, then we have discussed peptides, then we have discussed if we have a sequence, can we predict whether it can be a membrane protein or a non-membrane protein, because for a protein to stay in the membrane there has to be many hydrophobic amino acids come together to make that happen, right? And then we finally jumped into a discussion of membrane proteins and how many different types of membrane proteins present. Okay, so we're gonna continue from here in our next class, which will most likely be in the coming Tuesday itself. If there will be any change in the schedule, I'm gonna definitely share it with you guys. Chances are I might have one extra class this week or the coming week, I'm not certain, but rest assured I'm gonna share it with you guys so that you can have enough time for preparing your class, or at least be available for the class. Next, I'm gonna share the YouTube link to you guys via the mail itself of the playlist. I have not uploaded all the videos, but there are we have like more than 10 or 11 videos already been uploaded on that playlist. Okay, so thank you so much.